Hello everyone. So today we'll discuss about metabolisms and enzyme, which is in your Form 4 Biology Chapter 4. So the outline of today's video is that we're going to discuss about metabolisms and what is enzyme and what is the enzyme substrate complex and what is the enzyme nomenclature, how we name enzyme. And lastly, we'll discuss about the characteristics of enzyme. So before we start the lesson, let's ask yourself a few questions. So how many types of metabolisms are there in a cell? All right. And please list out 3 general characteristics of enzyme. I hope to answer your question in the end of this video. All right, so first, what is metabolism? So the definition of metabolism are all chemical reactions that occur in a living organisms. For example, conversion of food into energy in the form of ATP is considered a process of metabolism as well. But today, generally, we are going to discuss about 2 types of the metabolism in our cells, which is catabolisms and anabolism. So what is catabolism? Catabolisms, so when you see the C for catabolisms, they are a process of breaking down complex substances into a simple substances. So this process of catabolism is going to release energy. For example, like the breakdown of glucose during cellular respiration. So like in our body, human cells, um, we will break down the glucose to produce energy. So this process is known as catabolisms of glucose or breakdown of glucose. And next, on anabolism is a process of producing complex molecules from simple molecules. So this process of anabolism, you use or absorbs energy. For example, like glucose production during photosynthesis. So in plant, they will carry out photosynthesis every day. So they will use some substrates and to build the product, which is glucose here. And this process of building up something is known as anabolism. And catabolism is the process of breaking down something. All right. So this is for general. General, the catabolic reaction is A, which is a substrate. I will talk about substrate later. And then A substrate after being broken down or being catabolized, they will become B plus Cs, which are the products. And anabolic reaction, there are 2 substrate which they will kind of combine or build up or anabolize together and become C product. So these are very important reactions that you want to remember. So done with the metabolism. Next, we'll move on with what is enzyme. So enzyme is actually an organic catalyst. So a catalyst is something that's going to speed up or even fasten a process or any biochemical process in our body. So most of the enzyme are made up of protein, and the substance needed for an enzyme reaction is called a substrate. So substrate is something that will be that our enzyme is going to act on. So enzyme as a catalyst is going to fasten the process of a substrate, okay? And this substrate is going to bind with the enzyme at a specific site known as an active site. This is an active site is a site that we can find on enzyme, and then substrate bind to enzyme on active site to form a enzyme substrate complex. So you can see this is a substrate, and this is enzyme. And you can see this specific site here is very specific for this substrate and is called an active site. So they combine together and then they will form a enzyme substrate complex. And then the process of the substrate will be fastened or speed up by the enzyme. All right. Next, moving on to the enzyme nomenclature. So the name enzyme is derived by adding ASE to the name of the substrate. I will show you an example later. And this is by the International Union of Biochemistry and Molecular Biology for this principle of the enzyme nomenclature. So when you see here lactose is a kind of sugar that we can usually find in the, for example, like cow milks, okay? So lactose plus water, and then they will be catalyzed or fastened by an enzyme known as lactase. You can see after we change the OSE into ASE, a substrate will becomes an enzyme. Okay, So for example, now we have a maltose right here. So maltose is a kind of sugar as well. So maltose will be catalyzed by the enzyme maltase. Okay, so just change the OSE into ASE, and this will be the products glucose plus a galactose. All right, and then lastly of this video, I'm going to talk about the general characteristic of an enzyme. So the characteristic of enzyme is that these are a diagram that you want to remember. So enzymes is a protein or catalyst that they act very fast, they act rapidly. And then enzymes, they are used in small quantities and they are reusable. So for example, after the enzyme acting on the substrate, and then that enzyme will not like just gone, the enzyme can actually be reused again. Okay. And then enzymes, they will need a core factor to work. And then they also be destroyed by an inhibitor. So they need a core factor and they can be destroyed by an inhibitor. And the structures of an enzyme is unchanged or not destroyed after the reactions. Okay, so they remains there, and they are allowed to be reused. And enzymes, most of the actions of enzymes on the substrates, they are reversible. So for example, like the process that we mentioned just now, lactose, they'll acted by the enzyme lactase and it will becomes glucose plus galactose, and this process is reversible. So that means glucose plus galactose can become back lactose again. So this is known as a process that is reversible. And then most of the enzymes is very specific. Specific means that that enzyme only acted for that substrate. For example, lactase, the enzyme is specific or just can be used for lactose. Lactase cannot be used for glucose. Lactase only can be used for its substrate, which is a lactose. That's called specific. And this is the function of enzyme, which is to speed up biochemical reactions. So that's all for the lesson. To recap our video today, we actually talk about the definition of metabolism, which we have for anabolism and also catabolism. And we also describe the definition of enzyme, which is the catalyst to speed up any biochemical process. And we also talk about the enzyme substrate complex, which they will be acting on the enzyme active site to become a complex. And we also learn on how to name an enzyme by changing the OSE into the ASE, like lactose into lactase. So lactase is the enzyme. And lastly, we also talk about 8 of the general characteristics for enzyme, which we have, they are very rapid and they speed up process and they need a core factor and etc. All right. So I hope you learned something from this video. Thank you very much. And please like this video and comment if you have any question or any topic that you would like to learn in the future. Thank you.